Hello, everyone. I hear we have a full house. Is that great news or what? I mean, golly. Okay, so let me begin my thing. I'm Polly Armour, and I'm the research coordinator right now for the FDRS. And I'm going to talk to you a little bit about research, kind of just explain what the research process is and how it relates to the Gait study that we did last year. Who was at the conference last year? Okay, how many of you participated in the on-site research? All right, so we want to give you an update. It's not results really yet because we don't have any, but I'll let you know where we stand. Okay, so here are my disclosures. I work with Dr. Stutz. What's meant by research? We all have seen movies like this or cartoons, you know, the mad scientist with the wild hair, which is usually what I look like by the end of the weekend. It throws a bunch of things in a beaker to see what happens, boom, and no, that's not what research is. Research is much, much, much more complicated and hopefully less dangerous than that. It's a process, and it's a long one. And what you're trying to do is discover things that are already there, okay? You want to discover the truth, because just like in the, what is it, the X-Files, the truth is out there. It really is, and research does that mostly by eliminating wrong answers. So it's kind of like a Sudoku if you've done them. If you've figured out that this particular square cannot be 1, 2, 3, 4, 5, 6, or 7, then it must be 9. And that's a lot of what research is doing, is trying to eliminate wrong answers. It does this through a process called the scientific method. It's a 7 or 8 or 9-step process. There are lots of little loops. This is a simplification of it. But the first part of it is you ask a question that, like for me, I observed what was going on with my own body after I had liposuction, why my walking was so much easier even though my weight on the scale did not change, okay? Suddenly, I could walk for miles. And that made me think, what exactly is going on? And so I started to wonder, okay, well, how can we prove this? So what would be the question we would ask? Do people with lipedema walk differently from those who don't? And is this related more to body shape? Because one thing that happened after my surgery was my legs got a lot slimmer. Or is it more to BMI? Because remember, my weight didn't change and I didn't grow, so my BMI was the same. All right? The next step you do when you're doing research is you go gather all the information you can find on it. So you review literature, you talk to people in the field, you get together your collaborators and your equipment, all that sort of thing. And that was Karen Herbst and the treat people. It was FDRS, who was willing to do research here at the conference. Shelley came and added her Vanderbilt portion to it. Lipedema Foundation has kind of been helping with resources, all kinds of things. Those are the collaborators. It's a really kind of team approach that we're trying here. Then you have to form a hypothesis, and it's best if you can come up with a yes or no question that you can either prove or disprove. So the hypothesis for the Gait study was that women with adipose tissue disorders walk differently from unaffected women. And we can measure this, and we can see if it correlates with body composition, how the fat is distributed and how we're shaped rather than our BMI. So that was what we were trying to prove. So we performed an experiment, and in human research in particular, experiments are usually called studies. There's a bunch of different kinds of studies out there. There's observational versus interventional. So with an observational one, obviously you observe. With an intervention, you do something to the person and see if it makes a difference. Then there's also longitudinal versus case control. Longitudinal is when you observe people over time. Case control is when you compare affected people with unaffected people. So the Gait study was observational because we were just taking one set of measurements from each person, and it was case control because we were trying to match affected people with unaffected. Okay, most research, there's all kinds of different ways of doing research. Most of them fall into these types of categories here. Clinical case studies, non-randomized small studies, large randomized studies, literature reviews, and meta-analysis. And I'm just going to explain each of these and give you an example from the world of adipose tissue disorders. So this is a case study, okay? 1 or 2 people usually, sometimes 3 or 4, successful treatment of Derkham's by transcutaneous electrical stimulation. This just had 1 patient, okay? And you can find these things on the internet. They're not random because you're just looking at 1 or 2 patients, trying something on them. It appears to work with them. It doesn't necessarily work on everybody with that disorder, okay? So that's one of the limitations of case studies. Small non-randomized studies. So here's someone who took some tissue samples from lipomas and he compared them with normal adipose tissue. So this would be a case control study because he's looking at affected people and unaffected, but it would not be longitudinal, so therefore it's just observational. And then here, this is a non-randomized study. This is from Dr. Raprick in Germany and his colleagues. This is an interventional one. They actually did something to the people, and they tracked them over time, so it's a longitudinal study. Still, only 25 patients, not a lot, but I mean more than none. That's good. Okay, then there's the large randomized clinical studies, and these are the ones you sometimes read about in the paper. You know, they can have hundreds or thousands or even hundreds of thousands of participants. They're very often double-blinded. Okay, that's used a lot for drug studies and things like that, where neither the participants nor the researchers know when they look at the data. I mean, the participants don't know which drug they're getting, and the researchers don't know when they look at the data who had what. Okay, so that they haven't really done any of these yet, at least with our adipose tissue disorders. Literature reviews, you see these a lot. Somebody just goes in and collects all the papers they can on a disorder and then summarizes them in one paper. And this review of the literature, this has been around for a few years. It's actually a pretty good paper. And then meta-analyses, where you take the results of a whole bunch of related studies and combine them. So if someone were to take all the research that's come out of Germany with respect to liposuction for lipedema and try and combine them into one study, that would be a meta-analysis. There hasn't been much of that yet with subcutaneous tissue disorders. So now I'm going to talk a little bit about ethics. This is one of the things that drives research is the need for ethical oversight, okay? And this comes, there's a bunch of different statutes and I don't know what you would call them, things that affect this. Starts with the Nuremberg Code, which came out of the Nazi trials in the 40s after the war. It was later updated and expanded in the Declaration of Helsinki, and right now in the United States, they have something called the Common Rule, which is run, it's federal law, and it was most recently updated in January. That's the baseline standards to which all human research that's government funded in the United States has to be held to. And most institutions, even if the research isn't being federally funded, they expect you to follow these procedures. And most academic publications will not publish your research unless you have followed them. And if you ever need to sleep and you can't get to sleep, try reading the Common Rule. It will put you into la-la land in a moment. What this means is there's lots of paperwork and oversight, but that's not a bad thing because it leads to better research in the end. The main thing, though, is right on the top. You have to put, if you're doing human research, you have to put your subject's best interests first, okay? You can't take advantage of the volunteers because the volunteers are giving of their time to do the research. So I want to, whoa, that's the mic. As a researcher, I want to thank you all who have done this for giving your time to this. It's really, really important, and we appreciate it. Yay. So once you've gotten together all your data, you have to analyze it. There's a thing called data manipulation, which doesn't mean that we're fudging numbers to fit our hypothesis. It means we're going through it and putting it in a format that we can use to do the research. For example, last year at FDRS, we had 86 subjects come through, and there were 120,000 actual data points. So that's a lot of numbers and a lot of information to sort through and see what correlates. You have to check and double check and triple check. In one case, we had a participant whose BMI calculated out to 315. And I didn't remember anyone that would have been 315 for BMI. Turns out that the height and weight had gotten reversed, okay? And so it messed up the math. Well, we fixed it. Another thing is outliers. You have to check your data for outliers. That is people whose data is so outside of what seems to be the normal range that you think there's something else going on. For example, we had one person who took 20 very small steps to do that 15-foot walk during the Gait study. Obviously, she has some other thing going on that affects her mobility, so we take her information and we just set it aside. Then the other thing is, especially if you're doing case control, do you have enough subjects and you have enough controls, and do they match? Now, one of the issues we ran into last year was we didn't collect that many controls during the conference because we were so happy to have so many people with lipedema and Derkums. We thought, let's sample them more and just capture them while they're here, because we don't necessarily have that kind of access to folks. And we'll catch up with the controls later. So I went to Tucson in November and we collected a bunch of controls at the university there. The other issue is that you can have things that confound, that mess up your data. Like I mentioned the woman who took 20 steps. Other things, you know, are people who are old or young? Does that affect things? Are they obese? Are they very heavy versus very skinny people? The problem we ran into in Tucson, because we did the research, the captured controls at the university, they were young, slim college students. So we ended up with not a particularly good control group for them. Another thing to worry about is bias. A great amount of medical research is done in what are called weird countries: Western, educated, industrialized, rich, and democratic. Not that we're biased in the sense that we don't like people from countries that don't fit that. It's just richer countries tend to have the resources, the educated people, and so forth. So that's where the research happens here. The Gait study and the research we're doing this year, it's considered a convenient sample. So it's inherently a little biased because, first off, you have to know you have lipedema, so that tends to be people who are more severely affected. But you also still have to be able to get here. So the people who are really very significantly affected are not in this study also. So we're getting kind of the middle of it. Bias is not bad necessarily. You just have to know that you've got it and compensate for it as much as possible. Okay, so understand, it's not like bias, like I don't like people who can't make it here. It's just, it's the nature of research. It's hard to get rid of all of it. There's always going to be some things that affect you, okay? And then another thing to remember is, does measuring the thing change the thing? Okay, imagine if you were at the doctor's and they are measuring your height, and they want to make sure that your hair isn't too puffy, so they push down really hard on your head. You might end up appearing a little bit shorter than you really are. And another thing we've seen, and Dr. Herbst mentioned, the interstitium, right? It's all over the news, this new organ that they've only just discovered. The reason that they only just discovered it had to do with how they prepared the slides that they looked at under the various imaging things. And they have apparently been up until just now, they've been messing up the interstitium on the slides that they looked at, so they didn't see it, not because it wasn't there, but because they had damaged it during the preparation. So that's something really to be aware of. For example, when people walk on that carpet, no matter how many times I say, just walk like you're going to get the mail or you're going down this aisle at the supermarket, everyone is still thinking, I'm doing a gait study, I need to walk really carefully. Okay, that introduces a little bit of change. So we have to know about it and compensate for it. Another thing really, really important is don't draw the wrong conclusions, okay? You can say it 100 times. Correlation does not equal causation. That's really important. Also, you know, for example, touching on what Dr. Bartholomew said, he said 100% of people who use marijuana die. Also remember, 100% of people who don't use marijuana also die. If you ever go to this guy's website, Tyler Vigen, you can find he just collects data and then makes correlations with it. There are all kinds of fun stuff. It's like if you need to stay awake and you don't want to go to sleep, get yourself into this site because it's a rabbit hole if you're a data dork. So, for example, crude oil imports from Norway correlates almost perfectly with drivers killed in collisions with railway trains. Does not mean one causes the other, okay? Here's another one. If you're a winning, like, pay attention to the spelling bee every year because that will tell you how many people will be killed by venomous spiders. No, it won't. They just happen to come together. And this is an example of a negative correlation. This is my favorite. The number of films Nicolas Cage has appeared in is inversely correlated with people dying in helicopter accidents. But if you've ever watched a Nicolas Cage movie, you'd think it would be directly correlated. I don't know, whatever, you know. This is the exception that proves the rule, right? So one of the last things you do is interpret the data. Even if the data has no errors, you can still reach the wrong conclusion, okay? For example, there was some research come in the last 10 years, I guess, about asparagus and smelly pee. If you eat asparagus, does your pee smell like asparagus afterwards? And someone did some early research. They thought it was a genetic trait that the peers had, that certain people, yes, thank you, eat asparagus, they secrete something in their urine that's smelly. But later research disproved this, and it turns out that the genetic trait is not whether you smell, it's the ability to smell that strange metabolite in pee that is the one that's transferred genetically. So that's like the data was the same, it was just someone came to the wrong conclusion. The last thing is publish your results. For that, you have to write the paper, and you have to get all your ducks in a row. There's more information. Follow this website. It'll tell you all about the publishing, but we're not there yet, but I want you all to know that we are working on it, okay? So thank you all for your attention.